Just a moment. Okay. So we'll begin. I want to make the best use of the time. So you tell me, would you rather, what I've done in the previous lectures this week for this is do more of the past information since we're further away from that. Just a brief review of the first chapter, of course, and then looking at the chemistry is where a lot of people wanted to go back to. And then a little less time on the recent stuff since we've done so much on it with membrane and then the organelles now. So probably about 5-10 minutes on new stuff, with the majority of time will be spent looking at chapters 1 and 2. Is that good with you guys?
Yes.
Yes.
Okay, so hopefully by now, even just mentioning chapter 3, we're well familiar with the cell membrane, phospholipid bilayer, all that it means to have a fluid mosaic, meaning so many different things being put in that membrane, making it a mosaic, mainly these membrane proteins, of course, that give us so many things, so many opportunities to the cell membrane for transport and cell recognition, all that kind of thing, receptors. So I need you guys to, I guess, let me know before I go into the older stuff. Everyone feeling good with that structure of cell membrane? We have a great exercise in lab that has you label that. And then, of course, all the organelles. I mean, my goodness, there's not much time needed to be spent on that because it's the same old thing from high school. You just identify it and you label it, or you call out its function. So the organelles are got to be the easiest part of this whole exam.
Do we need to know the functions of the organelles?
Yeah, the functions, right. And I also sent you a review handout, like a 4-page handout showing you a picture of the cell and then a one slide showing all the functions. And if you haven't had lab yet, we'll be doing that in lab as well. Yeah, okay. So let's go back to chapter 1 real quick and look at body positions and directions. So simply put, body directions, that's talking about, the supine and prone. Which one is face down or laying on your stomach? That would be the prone. Right? Face up is supine. All the directions that I'm talking about here, the directional terms would indicate anterior, posterior, medial, lateral, proximal, distal. So we'll look at that briefly in the PowerPoint. The body planes, of course, sagittal, transverse, and coronal, knowing those 3 planes, and the oblique plane is a diagonal plane. But those first 3, what sections of the body do you get out of it? So we'll see like a sagittal section divides the body into right and left sections. A transverse or cross section divides it into upper and lower or superior, inferior sections. So that kind of thing. So hopefully all that's ringing a bell. The biggest thing coming out of chapter 1 physiologically was those feedback loops. So we're going to look at the 3 components that make up every feedback loop: the receptor, control center, and effector. So this is just to buzz your brain, just to get it all started up. And then all the organs, what organ belongs to which system, and getting to know brief functions for each of those 11 body systems: the skin, the bone, the nerve, endocrine, lymphatic, respiratory, reproductive, digestive, all those things. Knowing what organs belong to which system, and then having a brief idea of what each system can do. Both really for lab and lecture, you should know that, because you could get identification for either of those, right? Any of the organs being asked what quadrant are they in, or what region are they in, that kind of thing. All right, and then we'll get into chapter 2, looking briefly back at the atom and talk about electrons and the protons and such. And the bonding, of course, that's when we start. That's what the introduction was into our conversation of polar and nonpolar, is when we started talking about these types of covalent bonding, being a nonpolar or a polar covalent bond, which dictates the behavior of that molecule. So yeah, and then the water, of course, being a polar molecule, can't miss that. These are just the highlights before we jump straight in. Organic compounds, knowing your proteins, carbs, and lipids, what do they break down to become? So they start as a polymer, that's the protein, carb, or lipid in its biggest form, and they break down to become monomers, subunits, which is going to be the amino acids for proteins, monosaccharides for carbs, and glycerol fatty acids for the lipids. So all this should sound really, really familiar, getting to know how they're built up, broken down with the synthesis and hydrolysis, and then we'll finish up with acids and bases. Give me just a moment. Let me get that queued up. All right. So here's Mickey. Sorry about the little delay. My daughter was just texting me. She just got out of the dentist, having like 3 or 4 wisdom teeth being pulled out. So she's all a little bit loopy. All right. So here we have Mickey again. Remember Mickey when everything was nice and friendly? Now Mickey seems like a dragon. All these crazy terms that we've been pulled into just in a short few weeks, getting ready for our first test. But incidentally, the test is 100 questions, just so everyone knows. 100 questions, 1 minute per question. So that puts it at 100 minutes total. So if you're coming to campus, please allow yourself plenty of time to get that done. And conversely, if you're doing that on honor lock, realize that you can't, if the test is due at 11:59 on the 15th, which is Monday, you can't start it at 11 and finish it after 12. It's not going to work out. It's not going to let you submit it. You have to have it completed by 11:59. So I know that's cutting it tight. Please don't cut it tight with honor lock. That's a horrible idea. Don't do anything past 9:30, of course. You know, give yourself plenty of elbow room there. For technical glitches, make sure you got your cord plugged in. Make sure your camera's all working. Make sure you're in front of your camera. Can't leave that computer if you're doing honor lock. You just can't leave it. You could be tagged for academic integrity violation. So that's the risk with honor lock. But otherwise, we'll get going. Any first questions before we start into the pictures and the PowerPoint? Okay. So yeah, this stuff seems pretty friendly coming back, you know, looking at planes and such, and man, that's easy stuff. Looking at anatomy and physio real quick. Anatomy is the structure. Physiology is definitely the function. So we see many examples by now how structure determines function. One of the biggest, certainly coming out of our lab this week, looking at cystic fibrosis, the structure is all due to a dysfunctional chloride pump. Therefore, the function will not work. It won't pump chloride. So yeah, the structure of a DNA problem, not knowing how to build the chloride pump effectively, turns into a huge functional problem because the mucus becomes too thick in the respiratory passages because of the lack of chloride pumping out into the extracellular fluid, which would naturally attract the sodium and then the water, making the mucus a little more dilute, which is what we want. So that's a great example of that. Another example you could say would be the muscle fibers. If you got a tear in it, like a rotator cuff tear, well, guess what? That muscle's not going to work if you tear the muscle fibers. So there you go. You can't move your arm for a while with a rotator cuff tear. That's no fun, that's for sure. Looking at this picture real fast, a couple of these give us some great idea of a certain cut or a slice in a plane of the body. So at the top, bottom left, medical imaging, what type of plane would this be described? What kind of cut is that? Anybody know? Bottom left?
Transverse.
Very good, transverse. Yeah, so we're seeing the lung fields right there and a little bit of the heart right there in the center. So this would be in a thoracic area if you were asked, what type of transverse cut could you see both lung fields? Well, think about it. Where's the lungs? That's in the thoracic area, right? So that might be a little stretch of a question. Up here above that, exploratory surgery, what kind of cut would that be?
Frontal?
Yeah, very good. Frontal or coronal. We just took barely just the skin off there. Let's play with those organs a little bit. Let me pull this up here. What organ is this right here?
Is that the liver?
Here's the liver, the really big one.
Oh,
Tucked under the liver is the gallbladder.
okay.
So what quadrant would we be in if we're doing that?
Right upper,
Upper right
quadrant?
quadrant.
Very good. So this would be the right upper quadrant. If we go over here and see this organ,
Your spleen, is that your spleen?
right here is the stomach.
Oh.
And even though organs can be not necessarily displaced, but just in every person, the organs, you know, they're in slight different sizes, maybe depending on the person's genetics, and slightly different position. You know, sometimes the organs are exactly flip-flop. So when the liver and gallbladder could be on the right and everything's turned around. But in this case, given the picture you see, which is what matters for a specific question, given the quadrants that are drawn, which are standard, but given the way you see it here in this picture, that stomach appears more so in the what? Which quadrant?
Left upper quadrant?
More so in the left upper. The spleen you guys were mentioning would always be tucked just under the ribs on the very far top left. So this, even though you can't really see it well in this picture, that's where the spleen would be always tucked under the ribs right there on the left upper quadrant. Of course, we have small intestines all in the center. Large intestines, you can barely see it, but it's coming around. You see that rim of fat, all those yellow tissue pieces there? That's covering that large intestine coming up, across, and then down again right here. All these organs we've mentioned, what system do they belong to?
Digestive.
Right, digestive. So be aware of those kind of questions coming at you. It's going to ask you to either identify the organ or identify the quadrant it's in, or maybe one of those non-abdominal pelvic regions, or it can ask you what system does it belong to. Very, very good. Okay, let me just clear that out. That's a good start. Now, you guys should be thankful the way we changed it this semester. We usually include tissues on our first exam, which if you've taken A&P before, the tissue is probably everyone's least fun chapter, that's for sure. And that's probably comes because it's at the end of everything after we've done all this transport stuff. Your brain's usually full, can't fit any more tissue information in there. So I'm happy with the way they've divided it now, the tissues being on the next test. I think that's good to start off with. Okay. So the history stuff, I do want to ask you to make it a point to read through that in the book. Look for those highlight and bolded words in the book, like for inductive reasoning and theories and all that kind of thing. You definitely still want to read that. I just don't make it a point to discuss it in lecture because it's quite boring. But you could see a couple of test questions there in that history information. So make sure you do review that, just to be in all honesty. Okay. All right, our other big concept, because I'm just covering the highlights here. Tissues, organ systems all the way down to atoms describes the most complex to the least complex. So for this complexity question, the hierarchy, understand we start in the chemistry world down here in the atoms and molecules, and then we build up to the most complex here. So depending on how the question is asked, if you were to ask to put the hierarchy from most to least, you'd be coming here from the top all the way down to the bottom, putting them in order. But if you're asked least complex to most complex, you'd be starting here in the chemistry world. Even though it is complex, it's least or less complex than the whole body together. So yeah, atoms build up together to become molecules, or macromolecules is what the book says. Same old thing. It's just that macromolecules are really, really big molecules, like those organic compounds. That's what a macromolecule would be. And that builds up to become the organelles. The organelles group up to become cells. Cells group to become tissues. Tissues group to become organs. And then all those organs come together across those 11 body systems to give us the 11 body systems that we work on. all through AMP 1 and 2. 4 of which we cover in AMP 1, which would be the skin, bone, muscle, and nerve. And that's just showing you the same picture. The chemistry world down here, atoms, molecules, macromolecules being an organic compound, for instance, like DNA, but the other one's lipids, carbs, and proteins, just to cover that. It's building up to become organelles, and then cells, tissues, organs, and the system. Okay. Metabolism, another big concept we got out of chapter 1. Strictly put, it's the sum of all chemical reactions inside the cell. As long as you go with that definition, you should be just fine. It's the blending of the anabolism, which is the building up, and the catabolism, breaking down. So we covered that a few times, I know. That brought us into homeostasis, because of that metabolism, it's that strive toward optimum physiology. So it's not a stable, static internal environment, it's a dynamic internal environment, which means it's constantly changing, always, 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 reaching what it wants to have equilibrium, but always changing in the pursuit of that. So homeostasis is a dynamic. Relatively stable is a very, I guess, weak kind of way of describing it, but that's the way the book does it. Relatively, oh, it's relative, all right. It's conditional upon all kinds of transport mechanisms, isn't it? Our environment, our mood, what we ate, all the things that could change our physiology at a moment's notice. That's what homeostasis is fighting against, always. Okay, then we get straight into feedback loops. This allows for that dynamic equilibrium that we were talking about for homeostasis. It is not really necessarily an actual point that you get to with homeostasis, it's a range. So negative and positive feedback keep you in that range to be best explained. And negative feedback is designed to negate or reverse the original stimulus. So for instance, if you have a physiological condition with high thyroid hormone, or let's just say low thyroid hormone, then the body's going to attempt to make it higher. Or if it starts high, the body's going to attempt to make it lower. Or if you have low insulin, the body's going to attempt to make it higher. If you have high insulin, the body's going to attempt to make it lower. Those are all negative feedback loops. If you're cold, the body's going to attempt to make you warmer. Therefore, it makes you shiver and get warm. That's negative feedback. If you're warm or hot and you want to cool down, the body's going to make you sweat, which makes you cool down. Again, each of these examples reverses the original state that you were in when you started. That is negative feedback. When you reverse the original stimulus or state that the body was in. So here's those examples again with being hot and cold. We saw a few pictures there. If it's hot, then the body the room cools down with the thermostat. That's negative feedback. So there's so many examples. There's thousands you could say. That's why the best way to know this is that if you start one way and you end up the other, that is negative feedback. If you start warm and end up cool, negative feedback. For the case of this homeostasis with the compensation of blood pressure, check it out. Blood pressure drops here, doesn't it, in the beginning? And then it rises. That right there, my friend, is negative feedback. When you reverse the original stimulus or the original state that the body was in, negative feedback. And here's the control mechanism that does that. Please know these 3 words. You have to know these specific words. Receptor, control center, effector. That's what gets things done. In all feedback loops, not just negative. All feedback loops have a receptor, then the brain controls what that information that's coming in and then sends out another effect. So we're looking at cause and effect, but it's just written in this way. The cause is recognized by the receptor, of course. It sends it to the brain, and the brain sends out a message to fix it. Okay, so for positive feedback, we had childbirth and blood clotting, because this is where things go in the same direction. We get an initial contraction, and positive feedback dictates that we're just going to reinforce or, encourage that same stimulus, right? For more contractions instead of cutting them off, which would be negative feedback. And blood clotting the same way. We want more blood clotting. Once it starts, we don't want to kill that clot. So we want to keep the clotting going. Positive feedback. That's a reinforcement. We can skip these slides here. Getting into our body planes and positions, yeah, the 3 planes we talked about, sagittal, splitting the body left and right like this. If it's in the middle, it's mid-sagittal. If it's off to one side or the other, it's parasagittal. So if we're just cutting the shoulder off, that would be parasagittal. But right in the middle where it is right here is mid-sagittal. Coronal coming across the body like this, always in that way. Could also be called the frontal plane, giving us anterior, posterior sections, cutting the body like that. For transverse, certainly not just at the waist, it could be anywhere in the body, but that gives us upper and lower sections. I like to say sections because it's not always a half. And that is also known as the horizontal or cross section, that transverse plane there. So anatomical position, easy enough. Face forward, palms forward, legs apart. Yeah, we went over the idea that the arms are at the sides versus away from the sides. And I'll tell you what, just to be safe for the sake of your test, know it as it mentions it in the book as the arms at the sides. If it's a big problem on the test, I'll just throw the question out. But when you talk proximal distal, you know, the hand and wrist being the distal area, the shoulder being proximal, imagine that arm being out about 45 degrees rather than next to the body. That's been the more accurate depiction of anatomical position ever since I've studied it in all kinds of schools. So I'm not sure why they're updating it here to be at the sides, but if that's how it is on the test, just recognize at the sides. Okay, just go with that. And if it's a problem, like I said, I'll throw it out. Okay, there's your planes again. A good example of a frontal like we saw. Sagittal is the only one that can give you that medial flat edge where you can see the medial edge of something. Only can be viewed in a sagittal plane. And then the top here, viewed with a transverse. We're looking at the bottom half here. We've taken the top half of the brain off. So that would be a transverse or cross section. So get used to seeing examples like that. That's how they're going to ask you in lab what section is what, not just by definition, which would be more of a lecture thing, but in lab, they like to show you pictures and have you figure that out. Real quick on the quadrants and regions, just know how to play with those regions, knowing that epigastric is superior to umbilical, lumbar is lateral to umbilical, hypogastric would be inferior to umbilical, for example. Those just kind of relate it like that. And we went over the organs already, so that's great. You got all that straight. So don't get your right and lefts mixed up, of course. It's always the patient's right. Never, ever your right and left. It's always the patient's right and left. For the cavities, we use the division of the spine, giving us ventral cavities here in the front, dorsal cavities here in the back. So thoracic, there's some subdivisions there, plural for each lung, pericardial for the heart, and then mediastinum above that for the tracheoesophagus. And then all the abdominal organs, of course, in the abdomen region. So we use that worksheet we did in lab where we listed where each organ was placed in these cavities. Then I'll look at membranes real fast to get through that. Oh yeah, one more thing. All these terms here, yes, you're you know, anything could show up, any term. So we gave you about 10 or 12 in lab that we see the most, you know, common things that we talk about, but you would be responsible for any of these terms to be labeled, any of these regions on the body. For membranes, real quick, and that kind of does us in for chapter 1. Looking at only the serous membranes for right now, they give us a visceral and parietal layer. The visceral layer always wraps that organ, and parietal layer lines the inside cavity wall. So I mentioned this example, I'll do it again. If you think of yourself maybe having a bowl, okay? And you put a plastic bag, like a Ziploc bag, inside of that bowl, right? You wrap the inside of that bowl with a plastic Ziploc bag. Then you take a piece of fruit, like an apple or a cantaloupe or something, and you wrap that piece of fruit in saran wrap. Okay, so the wrapping around the fruit itself, the cantaloupe or the apple in this case, would be the visceral layer. It's the wrapping around the organ itself. Now you take that organ and you put it in that bowl that's lined with that plastic Ziploc bag, and that Ziploc bag would represent the parietal layer, lining the cavity that the organ sits in. Does that make sense? So visceral is always around the organ. Parietal is always lining that cavity wall that it sits in on the inside, whereas the visceral layer wraps the outside of the organ. So that's what can get people, but get that straight. And this is just showing where those things are. So the 3 words that we should study and know really well, coming up on this review slide here, right on there. Pleura, pericardium, and peritoneum. So know it by area and know which layer covers what or lines what. I mean, they're all linings, but get straight how to label these or describe where they're located. Okay. Excellent. So we've got about, what, 15 minutes remaining? Okay. Let's do some chemistry stuff real fast, and we'll maybe have 5 minutes for chapter 3 stuff, because there's a couple things I want to tighten up in chapter 3 that we've not spent too much time on. Oh yeah, there's your organs and body systems, by the way. Know how to match each organ with its body system, ending up chapter 1. Okay, let me pull up chapter 2. Any real quick questions on that? All right. For chemistry, we're going to go straight to the anatomy of the atom. Oh yeah, this is always a popular question, by the way. Let me go back to that one. Knowing what speeds up a reaction. Certainly a higher temperature, a smaller particle is faster, and a higher concentration. And of course, enzymes always speed up reactions without being used in the process. So know that slide. That's always a popular question all around the world on a first RMP test. Know these big players here, these 4, making up 96% of the body: carbon, hydrogen, oxygen, nitrogen. And then the anatomy of the atom real quick. Only the protons and neutrons are in the nucleus. Electrons are always in these other shells. So how do we account for the mass of the whole atom? Because the electrons are not really considered in the mass. We just weigh what's in the, or consider what's in the nucleus. So what's the atomic mass or mass number here? How many particles do we have?
4.
In that one. It's 4. Only 2 protons, 2 neutrons. That gives us 4, which gives us the mass number. The atomic number is only proteins, remember, or protons, excuse me. So the proton count is always the atomic number, same old thing. And then for the math of that, we can figure out neutron count based on the atomic mass and then subtracting. So we'll see an example of that real fast. There's your mass number, which would be nucleus, excuse me, with the protons and the neutrons together. So let me give you a quick example on that. If I give you a mass number of 14, and I say that there's atomic number of 10, how many neutrons would there be?
4.
You would just take the larger number and then subtract that smaller number, and you're left with the neutron count. That's simply it. So a 14 mass number is everything, protons and neutrons. If I tell you 10 is the atomic number, which is the protons, then you're just left with 4. So that must be the neutron count. There you go. So isotopes just mean different versions of the same atom, with the neutrons being different, which gives you the atomic weight to account for different weights of each of these, right? All averaged up together. So in summary, know how to play with these 3 words. You'll get examples of questions thrown at you with just the atomic number, which is the same as the proton and the same as the electron count, to tell you the truth, unless the statement stated that it lost or gained an electron. Stable state would have the proton count equal the electron count. So please review those notes. Mass number, of course, or the atomic mass, gives you both protons and neutrons. And if we're trying to figure out something, another example, if I give you a mass number of 28, right? And I told you that 14 was the proton count or the atomic number, then you're left with another 14, which would be the neutrons, just to make it real easy. That's the only math you'll have to figure out stuff like that. Electrons are negative, and they give us the ability for things to bond together. That's what's important. Otherwise, when electrons are gained or lost, they give us ions. So a cation means that you lost an electron, It's positively charged, so it'll always attract a negative charge. An anion is negative because it gained another electron. It'll always attract a positive charge. So they always attract their opposite. And that takes us straight into bonding. Know your three types of bonds, of course, and examples of each. What's our classic example of an ionic bond? Sodium chloride for ionic bond. Examples of covalent bonding from polar would be water, of course, inside the water molecule. For nonpolar, it would be like gas, like O2, CO2, or methane gas. Not all gases are nonpolar, by the way, if I've misled you in that direction. Some gases are polar, usually the more dangerous ones. But the nonpolar gases that we cover are O2, CO2, and methane. So that's why we mention it so much like that. And hydrogen bond is strictly between different water molecules. Please get the difference. The polar covalent is within one water molecule. The hydrogen is between different water molecules. So there's methane, it's nonpolar, there's no charge. There's nitrogen gas, nonpolar, no charge. Carbon dioxide, nonpolar, no charge. Holding those three things together is nonpolar bonds. Holding a one water molecule together is a polar covalent bond. So remember the definitions. Ionic is lost or gained, right, or transferred, whereas covalent bonding shows you a sharing of electrons. We have two types of sharing, equal and unequal. If it's unequal, it's polar. And if it's equal, it's nonpolar. Because polarity builds both charges, whereas nonpolar builds no charges. No charge, nonpolar. When it's unequal, you get both charges, negative and positive, which means water can want to bond to lots of other things that are polar, right? Because of attraction of the charges. And certainly it wants to bond with sodium chloride. So real quick, let's do that one. If sodium is the cation, what side of water will it be attracted to? If sodium is the cation, it's always going to be attracted to the negative side of water, which is the oxygen. And if chloride is negative, the anion, it's always going to be attracted to the hydrogen here of the water molecule. And that's how water disassociates sodium chloride. Perfect. There's our hydrogen bonding between the water molecules. Please get that straight. That also gives us our surface tension, which is also described as cohesion, water sticking to itself. Surface tension a little bit different though. It is the increased tension that occurs at the surface of water from the collective hydrogen bonds. So it's a little bit stronger than underneath the water. At the surface, it creates like a film. That's surface tension. That's the hydrogen bonding, by the way. Okay. Last part of this, everybody good with the hydrophilic, hydrophobic comparisons? Remember to match up the words there. Hydrophilic is everything that's polar or otherwise water soluble. Hydrophobic is nonpolar, lipid soluble. So we made really great distinctions on that, and which brings us straight into our transport conversation, because nonpolar molecules through a hydrophobic area will only allow things like lipids, steroids, and those nonpolar gases, right? Because hydrophobic and nonpolar molecules like to blend together. But for hydrophilic and polar, these polar molecules that we see like sugar and amino acids and ions, which have a charge, but they're not polar, anything with a charge cannot pass through that nonpolar fence of the cell membrane. It's going to need help in the way of a membrane protein. And then lastly, for this chapter, getting into the organic compounds and then the pH. This is material straight out of lab, so really just use your lab notes for that. Know how to break down each of these organic compounds to their monomer state. Monosaccharide here for carbs, proteins down to amino acids, and so forth. So get those monomers straight, and then understand how each are built up or broken down using dehydration synthesis to build up, or hydrolysis to be broken down. I want to try to save us some good 5 minutes here for our last stuff. For the acid base, watch that video one more time. I did include a few examples in here. Let me get to that real quickly. from the video. If you've watched the video, that's great. It's not like it's fancy, but it does describe some good examples in there. And I've gone ahead and included these examples for the acid and bases right here. If you can understand this page in the updated PowerPoint, then you're going to be doing pretty good.
Dr.
So,
Sliman,
yes.
I think we're here till 10:45.
Oh, I'm so sorry. Thank you. Yeah, I was looking at that wrong. Yep, you are so correct. Thank you. I appreciate that. Yes, I so looked at that wrong. That's wonderful. Well, then we do have some elbow room. Wonderful. Well, yeah, let's talk about then about the structure of proteins, the fibrous versus the globular. That's very important because it gives you a lot of things we've seen already, like the microtubules, microfilaments. Let me go back to that slide even. I don't know what's wrong with my brain today. I'm so upset with my computer, guys. It gave me all kinds of weird things this morning, scary things. I was afraid I wasn't going to even be able to open the session, but thank God it finally came around. Here we go. Okay. Yeah, these two types of proteins, in bold, please know these examples. We see those cytoskeletal filaments coming from the fibrous category here, being more like fibers, whereas the membrane proteins are certainly globular. So please know those examples. Recognize the denaturation of proteins. That's when some of the attraction and the bonding of the proteins is lost. So yeah, be aware of that term, denaturation for proteins. Whoops. And what happens when you change the structure of something, by the way? When you change the structure of something, you change the function, right? So a denatured protein not only structurally is not together, but functionally it loses its functions. It's deactivated, basically. All right. One more time about enzymes. They speed up, but they're not used in the reaction. That's great. Let me actually go back now that we've got the time straight and we're back in the normal world. Let me go back to that. Thank you for your patience with that. Let's go back one more. There we go, right there. Since we have a moment now, I do want to pull something up. Give me just a sec. Yeah, I think I flashed back to 2015 there for a second, when all of our lecture classes were, you know, like that. You know, put something up on here on the whiteboard, and I want you guys to study this in this way. It'll help you get straight the information that we need to have for this hydrophilic hydrophobic story when you compare it to polar nonpolar. Okay, so if you would make a side-by-side like this so we can get straight some word association. So when we think of things that are polar, we definitely know that it is, well, let me do it like this. Water soluble. Water soluble means, yes, it could be water itself, or it could be another water soluble compound like sugars, amino acids, or it could be anything with a charge. That just means it's water soluble. So I'm saying that I'm saying that polar is attracted to other water soluble compounds or ions. So it's a word association, it's an attraction. Whereas nonpolar, what is it known as? It's known as lipid soluble, but what the same of saying lipid soluble is water insoluble. So I want you to do this because it makes you practice that big game of opposites that we talk about so much. So if polar is water soluble, then nonpolar would have to be water insoluble. But yes, nonpolar is lipid soluble, like we mentioned, which would make polar lipid insoluble. So this is just to show you the opposites, how things can be worded. Now with that comes those two terms we just saw. Polar is going to be hydrophilic. Whereas nonpolar molecules, hydrophobic. Meaning they do not like to mix with water or polar molecules. But they do mix with each other. So hydrophilic and hydrophilic go well together. Hydrophobic and phobic go well together, but not the two compared to each other. Okay, so with this builds the whole idea of transport. Which one of these words, polar and nonpolar, will describe the molecules that use simple diffusion?
Non-polar.
There you go. Simple diffusion will be under the nonpolar area. Very good. And why is that? Why do we call it simple? Because that cell membrane is nonpolar. I don't know how many times I can say it. It does have a polar aspect to it at the phosphate head, but so much of it is nonpolar that we considered its function to be more nonpolar. Since 90% of it is nonpolar, then we're going to call it nonpolar, which means it's going to use simple diffusion. Nonpolar molecules will use simple diffusion. That's what I'm trying to say. Or they simply go right through without the help of a membrane protein. Now, if you've got room on your sheet, I don't, but underneath simple diffusion, right, maybe a couple of dashes, right, underneath it, so that you know things like lipids, steroids, and nonpolar gases like O2, CO2. That's what uses simple diffusion. Lipids, steroids, and O2, CO2. And methane, if you want to throw that in there. Lipids, steroids, and nonpolar gases like O2, CO2. Okay, what about on the polar side? What kind of diffusion do we use there?
Facilitated.
There you go. You got to have all that straight before the test, no doubt. And things that will move with facilitated, write yourself a couple of dashes underneath for examples, would be sugars, amino acids, and ions. And of course, water itself using osmosis, to be honest. Sugars, amino acids, and ions. And water itself. Underneath the nonpolar category for simple, you can include fat-soluble vitamins, if you want another example. Okay, great. I find those side-by-sides to be extremely useful because they get the word association real nice and tight for you in the brain. All right, wonderful. Back to this stuff here. Any questions on that side-by-side information? Did y'all get all that? Let me drag it back up real fast, maybe take a picture of that. It's not very neat. So hopefully you're writing it yourself in a neater way. But these are the words that are associated together that will help you out with some good test questions, I'm sure. Okay. All right, so know how to play with these words, hydrolysis versus synthesis. Which one of these builds up compounds from small to big?
Dehydration synthesis.
The synthesis does, right? This is an anabolic pathway. It builds up things. So know how to recognize that by definition, of course, like we just said, but also recognize how it's written, where two small things build up to become a bigger thing. That's dehydration synthesis, water being lost in the process, yes, but we're still synthesizing something. That water being given off just means it makes a bond happen between A and B. Here, when we use water to break things down, it looks like this. This would be a decomposition reaction where we start with something big and then we break it down into smaller units. That's hydrolysis. So for instance, a protein being broken down from the macro level to the micro level, right? Proteins from big to small would be hydrolysis, down to the amino acid level here. If it were a carb, it'd be down to the monosaccharide level there. So the carb and protein are always the macromolecules, right? The bigger ones, the polymers, as they say. And the monomer is the smallest unit. After you break a polymer down, you get monomers eventually to the smallest unit. Okay, so again, for the organic compounds, just study those on your own, know what the monomers are for each of those. And we studied a little bit more about proteins. Know that lipids give you the most energy of all the organic compounds. Lipids do. Carbs give some energy, but lipids give the most. As we know, fat has lots of energy in it, lots of what you could say potential energy, right? All those hydrogens that are in fats, holy cow. Yep, that's a lot of energy. And there's your shapes again for proteins. Again, those examples we mentioned. Please know these examples for each of these categories. All right, let's get to it. Last part will be the acids and bases. Like I said, just look at that video for the acid and base review. Let's get down to that area there. The highlights for acids would be that they give off hydrogen ions. So the lower you are on the pH scale, the more hydrogen ions you give off. So if I say everything under 7 is considered an acid, then a pH of 5, yeah, that's acidic. But what about a pH of 2? It's a lower number, but it's more acidic. So please get that straight. That's the biggest confusion I find people have on this, is that the lower the number, the more acidic you're going to be. So a pH of 2 is more acidic than a pH of 5. Therefore, the pH of 2 will give off more hydrogen ions than a pH of 5 would. So that's what the video explains. The video also shows in the book that acids are considered proton donors. And in this case, of course, well, in all cases with acids, they give off that positive charge and then an excuse me, I can't even talk, an anion. For bases, they're known as proton acceptors, and they give off a cation. So hydroxide is going to be the key thing in bases, whereas we're looking at hydrogen for acids. So the more hydrogen ions that are given off, the more acidic, which would be the lowest number on the scale. The more hydroxide ions that are given off on the pH scale, the more basic it is. So comparing a base of 8, the number 8 on a pH scale, compared to a base of 11 on the pH scale, the base of 11 will give off more hydroxide. The base of 8 will give off less hydroxide. So that's what the video explains. I know I'm doing that kind of fast, but yeah, that needs to be something straight in your head as well. And they're known as proton acceptors. And here's these examples I'd like for you to know. Not the exact numbers, of course, but just know how to relate the numbers in terms of what's more acidic, what's more basic, what gives off more hydrogen, what gives off more hydroxide. That's what's important. Okay. And of course, the salt, no hydrogen or hydroxide, it's just a salt. It's not an acid or a base, it's in its own little category. All right, and then a buffer. I'm so sorry about that last slide there. A buffer helps prevent large changes in that acid-base relationship. So if you're too acidic, the body's going to want to put a buffer out to match that with some base to bring that acidity up. If you're too basic, the body's going to want to match that and keep some acidity in the body. And it does that through the kidneys, by the way, or through your breathing, so to speak. Yep. So that's a buffer situation. So next up, we get a little bit of time here on chapter 3 information. Any questions that we need to go back to really quick on the chemistry stuff? Okay, how are y'all feeling in general for the test? Feeling somewhat ready?
I felt good until you said it had 100 questions.
Yeah, it does have 100 questions. Yeah. And then 100 minutes. So 1 minute per question. Most questions you're going to be able to answer, of course, under 30 seconds. So it'll actually be enough time, believe it or not. A lot of questions you can answer under 10 seconds. And if you paid attention in lecture, if you're able to hold some kind of agreement in what's being stated, like at this point, everything doesn't sound so foreign, then you're prepared, right? If you know how to write things down on paper, if you know how to distinguish between active and passive transport, polar and nonpolar, philic and phobic, all these kind of things, you know, anterior, posterior, all the basic stuff, you're going to be well prepared. Getting the details straight, knowing how to bend it and be flexible with that information determines whether you get a B or an A. But if you know all the basics, you're definitely going to pass. Read the questions very carefully. They might be worded differently than what we're used to, but the content, the information is still the basic stuff there. So read carefully so you don't jump into an answer that may be incorrect. So we have some time to do some chapter 3 stuff, unless you guys have any questions on the old stuff. Well, that's great. Okay. So let me touch on a couple of things in chapter 3. And I did post videos, by the way, from our last lecture and our this week's lab. So those are posted if you want to review those. Once more for lab review, because really what I'm talking about here is mostly lecture, which does go into lab. But for the lab review for yourself, because roughly 20 to 25% of the test will be lab questions. My best advice, besides studying whatever models you might have seen in those application handouts or Connect that you would have labeled, would be to go over the handouts that you did. So the water worksheet for the chemistry lab has lots of great test questions in it. The worksheet we're doing this lab this week for the cell, great many amount of questions there. And then of course, all the transport stuff, you know, we've done a few review quizzes in class and lab. So use that as your review. So any handouts you might have gotten for lab, that is your study guide for lab, for sure, no doubt. Okay, for cells, oh my goodness, so much to say, isn't it? There's a review of our 3 important words for homeostasis: receptor, control, center, effector, controlling all those nice feedback loops. And here's just some examples of all that. We don't need to do that again. Remember that negative feedback loop, whoops, let me go back one, is reversed. Whereas a positive feedback loop, we want to reinforce or strengthen the original stimulus. So this just reviews that real fast in this part of the PowerPoint that I've done. Your hierarchy again from least to most, right? Get that straight. So if you went from most to least, you'd start at the organism down at the cell level. But if you work least to most, you start in the chemistry level and go all the way down to organism level. So pay attention to how that might be worded for your tests. Okay, let's get to the cell stuff straight out. Real quick on cell junctions, these are just special proteins that hold together cells from the plasma membrane, cell membrane itself. So do know these 3 types. A tight will be found in the intestines. Desmosome is found in the skin and heart. And gap junctions be found in the muscle to share electrical charges. That's how the heart muscle works together as a unit. So yeah, one of those 3, of course, will be thrown in there. I just want to get to the main review slide here. There we go. This slide starts the last PowerPoint video that was posted there, the part 2, looking at the overall summary qualities of that cell membrane, noticing all the membrane proteins embedded there, whether they're integral or peripheral. Of course, one more time, if you want to look at that. Anything going into the membrane itself would be integral. whether it's fully like this one or partially like that one. Anything right at the surface would be peripheral. Okay. Comparing active versus passive, downhill would be passive, uphill would be active, and active requires ATP, whereas passive does not. So please understand that one. Here's all those processes we've been through many times. These are all the versions of passive transport you can get. So yeah, simple is definitely non-polar, lipid soluble, right? Please get those terms straight. I've seen in lab, all three of my labs so far, there's still some mix up there. People want to say water is simple diffusion, and even though water can move with simple, it's more accidental, but water moves through osmosis, which is a type of facilitated. Water is not lipid soluble, so it does not move through simple diffusion on a great basis. It does only by small amounts and by accident. Facilitated movement is going to be your water soluble solutes like glucose, which is a sugar, amino acid, and or ions. Anything with a charge like that will move through facilitated. So that's why I took the time to write that side by side with you. I think it's very valuable that you do that same thing, study it, own it, work it on a separate sheet of paper so that you get all those words straight. They mean a lot. They represent a lot of points. that chart, I wouldn't have used valuable time like that without it being important. Please know that chart that we wrote down. Okay, so there's your examples right out of the PowerPoint. Nonpolar, gases like O2, CO2, steroids and lipids, that's what will move through the cell membrane simply, right through. No membrane protein needed, go straight through. And the polar world, that's for facilitated. Not just polar, but anything with a charge like an ion also moves with facilitated. That's where we get our sodium, potassium channels from. Not the pump, that's active transport, but the channels are the passive, which is facilitated. Anytime you mention a channel, by the way, or a carrier in that way, we're talking about facilitated. Now with sodium potassium pump, where they go, the sodium's out, the potassium's in, that was the pump. That's an antiport. That's two ions at the same time. Not at the same time, excuse me, but in the same pump. Whereas in facilitated, you're going to have a separate potassium channel and a separate sodium channel. With the active transport, they're moved through the same protein, which is the active transport sodium potassium pump. So realize that is a difference. And that one's called antiport, right? Antiport means two ions going in two different directions in the same pump. That's antiport. So we'll see that coming up. There's our beautiful aquaporin for osmosis, of course, only for water. Dedicated channel. Now, this is a slide I wanted to go back to because it's a big slide. It's got lots of info on it, and I find it to be a tough part in the chapter. But check out that newest definition I've added because it is the basic definition coming out of the book. I know we talked a lot about osmolarity, and it seems like it's all over the place, but it does represent the actual total number of solute particles that's measured per liter in a solution. So please make a note of that. Osmolarity, which is the same as osmotic pressure, is the total number of solute particles per liter in a solution. Or strictly put, the total number of solute particles. There you go. That's the osmolarity. Everything else that's described here just means that it's gonna, the more solute particles in a solution are gonna attract more water. That's what that functionally means. But to define it, it is the total number of solute particles, osmolarity. And then compared to tonicity, we see the tonicity in the same way, but just defined differently, the ability of that solution to affect the fluid volume. So when we talked about that red blood cell being put into a hyper or hypotonic solution, all we were talking about is how is water going to move, right? The red blood cell just provided a means compared to the concentration that was already there. So that's where the fluid volume changes. It's going to be decreased or increased depending on where that water moves in or out of the cell. So these definitions, I find they confuse a lot of people all the time, but I wanted to be clear about it. Please know these red bolded definitions. For osmolarity, it's total number of solutes. And then for tonicity, the ability of solution to affect fluid volume. Because the fluid's going to move in one direction or the other. If we put a red blood cell into a hypertonic solution, oh gosh, I put the wrong, let me switch that, my goodness. Let me fix that real fast, guys. Okay, so sorry about that. When you put it in a hypotonic solution, yeah, that cell, what's going to happen to the cell, of course, is that he goes through hemolysis. It's going to attract water. So it's going to take water from that fluid environment and bring it into the cell. That's what I mean by affect fluid volume. For the hypertonic side, it's going to shrink that cell, which means there's going to be more fluid volume added to the solution. So again, we're affecting the fluid volume of the solution by the tonicity that's there. So doesn't that sound confusing? It's a different way of looking at it, but that's how the book likes to describe it. So I thought it only proper that you know for the test how the book describes it. Functionally, what we've covered in lab is certainly correct. What we learned is where the water goes in respect to the bag, right? And it really just looking at the opposite side of the coin here when we introduce this other description. So in a hypertonic solution, yes, a red blood cell does shrink because it gives the water up to the solution, which affects the fluid volume of the solution. That's what it's, so you're really just looking at the other side of the coin. Instead of just thinking about the red blood cell the whole time, you think about how that fluid volume changes in the solution. So in other words, a hypertonic solution, since it has more solute, is naturally going to attract more water towards itself. I guess that's the equality of all. A hypertonic solution will attract more water to itself. A hypotonic solution will lose water. It'll go into the cell. So again, we're just talking about the flip side of the coin, and hopefully I didn't mess your brain up too much with that. Isotonic is just no net flow. Water moves both equally in and out. So all this same stuff is represented in lab. All of it, we do a nice review of everything. There's our active transport, 3 sodiums out, 2 potassiums in, giving us the major extracellular cation being sodium and the major intracellular cation being potassium. That should be easy to know by now. Okay. Getting past that stuff. Oh yeah, one real quick thing on the negative charge here on the inside of the membrane. That's probably the biggest takeaway you need to have for this resting membrane, because we do come to it later. So please understand when we say resting membrane, it's what makes the membrane polar altogether, electrically speaking. Where there's a net negative charge on the inside of the membrane because more ions are leaving the cell, which makes it negative on the inside overall, and more positive on the outside. So right now it's such a big story. Just understand this term right here. Net negative charge on the inside of the cell membrane is what a resting membrane potential is going to establish. And then that's described between all these events that go on here. Right, all these things, lots of words, that just describes how it achieves a net negative charge on the inside. This first one has to do with active transport. More sodium ions or more positive ions are being pushed out, less positives being pushed in. Therefore, you end up negative on the inside. And then passively, it's more permeable to potassium. So again, more ions go to the outside, leakingly through passive transport. Then proteins are trapped on the inside and they're negative. So again, more negative on the inside. But that's a big story, like I said. Getting to know all your organelles is the easiest part. I wanted to show you one more slide before we go. It's a picture of the lab model that we have, that you have in your worksheet this week in lab. Here it is right here. So yeah, can you see the likeness of the models that we use in lab for the different organelles? So if you've got another minute, I can just label these with you real fast. So of course, this will be the nucleus with the nucleolus inside. Again, the nucleus over here with the nucleolus embedded inside. In the nucleoplasm, which would be the inside area of the nucleus, that's all where the chromatin or DNA is. Over here will be right here, the DNA. The rough ER on this model here, and the rough ER on this model here. It just, this was like from 1947, so it looks different, but look at all the dots. That's rough ER. Whereas over here is smooth ER. And right there is smooth ER on this model. Mitochondria here on this model in a whole form and then a cut form. And a mitochondria here in a whole form and a cut form right there. Lysosome right there. Lysosome right here. There we go. The Golgi looking kind of strange right there. Golgi on this model here. Centrioles that live in a centrosome. That black body is a centrosome that just contains the centrioles. And then we have centrioles over here. So the structures look the same. They may be different colors, but that's all the basic stuff. You know, ribosomes, of course, can be bound to the ER, or they can be freely floating like these little jagged edges right here. But that's it. Yeah, that would be a peroxisome right there, a peroxisome right here. So the lysosomes are typically yellow, to be honest. Not that the yellow matters, but that's how they do it in the models. Okay, guys, I think that is it. And remember our passage of a protein. What 4 areas of the cell do we use for a passage of a protein? Number 1 would be the nucleus to get the instructions for the protein. Then we bring it to the rough ER to be synthesized and transported, and we transport it to the Golgi to be packaged. And then the Golgi uses these little vesicles to push it out of the cell. So nucleus, rough ER, Golgi, and then cell membrane. And that's also, those 4 stages are in your lab PowerPoint. Okay, guys, we did it. We made it to to the correct time of 10:45. If you have any questions, let me know in an email. I can. I'm happy to answer them, or anything quick right now. I'm fine. I got a few minutes.
Oh, I have a question.
Yes.
Is like the adduction and flexion going to be on the exam?
Oh, the muscle movements? No, that's not on this test.
Okay, I just wanted to make sure.
Yeah, sure.
Thank you.
Oh, you're welcome. Any other questions?
Thank you.
You're very welcome.
Thanks.
All right. Well, best of luck to everyone.